So wait. So very good afternoon, one and all, and I welcome you all in a ISFCP dialogue series. Today we have the speaker of the day, Dr. Jayant Kumar, who is currently working as an assistant professor in the Department of Pharmaceutical Science. He did his B Pharmacy from RGPV University, Bhopal, Madhya Pradesh, and he did his MS in Pharmacology and Toxicology from NACO Mohali, and PhD in Pharmacology and Toxicology from NACO Library. His area of research is basically focusing on neurotoxicity related to macrotoxicity and chelation. We welcome you, sir.
Thank you, ma'am. Hello. Hello.
Yes, sir. You are
May I share,
audible, sir.
ma'am?
You can present. Yes, sir.
Yes. Is it visible, ma'am?
Yes, sir.
So thank you, ma'am,
Uh,
for
yes,
inviting
sir.
me
It
at
is
this forum. So I'm going to discuss about the repositioning of an arsenic chelator for Wilson disease. So after looking this title, you might be thinking that why Wilson disease? Because it's a rare genetic disorder, and there are few patients in India, and the drug which is used in the management of Wilson disease is like depenicillamine, which is available in the Indian market. But let me tell you, the drug depenicillamine is not available in Indian market. So our because due to the short supply of raw material which exported from the China, so our ministry is looking for a new safe alternative of this depenicillamine. So that is why I have chosen this topic for my research. So let me tell you brief about the copper and its associated disorders. So like copper we are using since antiquity, and it has very remarkable role in human cellular physiology. It serves as a cofactor for many enzymes like uh, SOD, beta hydroxylase, and many enzymes. They control various functions in the body, but at the excess in the body it causes, or it is associated with the several neurological disorders like Wilson disease, Alzheimer disease, Huntington disease, ALS, prion disease. So the next question is how we exposed to copper, and copper, how copper contribute in the neurological disorders or in Wilson disease. So the major source of exposure is copper plumbing. So here is the report. After 1950s in US, more than 90% houses have the copper plumbing in their homes. For that reason, epidemiological studies linked Alzheimer disease with a higher copper level. Second is extensive use of these copper utensils. Third is copper-based agriculture products like copper oxychloride, which is well-known antifungal and antibacterial activity, and copper tea. These sources increases the body copper level. This copper absorbed from the intestine, and then it goes to the liver where it get processed and incorporate in the celluloplasmin, and then it distributed in throughout the body. Then it reaches to the blood in the brain where it bind with the amyloid beta protein, and in normal condition this amyloid beta protein helps in the clearance of this excess copper. But in the disease condition, or we can say at excess, it causes the precipitation of this amyloid beta protein. So here is a interesting study in which researcher isolate the amyloid plaque from the healthy subjects and the Alzheimer patient, and they got that the copper level was significantly higher in the Alzheimer patient's plaque as compared to the healthy subjects. Then these amyloid and copper plaque deposit on the neurons where it causes the reactive oxygen species generation. These reactive oxygen species generate the oxidative stress and leads to the caspase activation, which eventually leads to the neurotoxicity or neuronal cell death. So how you can see here that copper contribute in the neurological disorders, but in Wilson disease, which is a rare genetic disorders in which the Wilson disease, the major target of organ are liver and the brain. So in hepatic cells and neuronal cells, copper enter into the cell with the help of this HCTR, human copper transporter 1. Then it when it comes into the cells, it get processed and incorporate into the ceruloplasmin with the help of this ATP7B protein. But in the case of Wilson disease, this ATP7B protein got defected, and this whole process disturbed, and which leads to the excess buildup of copper in the cell, which eventually lead to the hepatic cell and neuronal cell death, and at last causes the organ failure. So here, researchers suggested that metal chelation therapy could be useful in the management of copper-induced neurological disorders and the Wilson disease. And these are the agents have been screened earlier, but they have been associated with the several adverse effects. So there is a need of new copper chelator which could be safe and effective against the copper toxicity. So we have a molecule MiADMSA, which is an indigenous molecule. This molecule was developed by DRD Gwalior, and if we talk about the its chemical profile, so it is a lipophilic in nature, and it's an analog of DMSA. So being in lipophilic nature, it can easily cross the blood-brain barrier and also a cell membrane, and it can remove the metals from inside the cell, not only from the blood circulation. It is effective. Initially it was effective against the arsenic poisoning. It was investigated against the arsenic poisoning, but later on it has been explored for the copper toxicity. It has completed phase 1 clinical trial. It has strong metal chelating and free radical scavenging property due to the presence of this thiol molecule. And during the preclinical investigation, it has been observed that it is also depleting the copper, essential copper. So we thought that if it is decreasing the essential copper level from the body, so could it be useful in the neurological disorders, copper induced neurological disorders? Or if it is decreasing the body copper level, could it be useful in the Wilson disease where copper level is increased in the organ? So to test our hypothesis, we designed these objective. So first, we characterize the animal models. Then we investigate the copper chelation activity of MiADMSA using the NMR and fluorescence spectroscopy. When we confirm that our drug MiADMSA is binding to the copper, then we perform the in vitro cell line studies to check its effect in vitro before going to the in vivo. Then we compare our drug effect with the D-penicillamine, which is a first-line treatment for copper toxicity. We have observed its effect on the brain and liver. So these are the methodologies that we have used. We used the animal model, Sprague Dolly rats, Neuro 2 cell line. We have used the chelation activity. We have measured with fluorescence and NMR spectroscopy. Parameters we have evaluated: the body weight, oxidative stress parameters, neurobehavioral parameters, histology, HNE staining, picrocerebral res staining for the assessment of protein marker. These markers we have evaluated by the qPCR and ELISA methods, and we also measure the copper level using the atomic absorption spectroscopy. So our first objective was to characterize the copper induced neurological disorders in Sprague Dolly rat model. So this was the study design for objective. First, we randomly divide animal into the three groups. Group first received the normal drinking water and serve as a control. Group second received 10 mg per kg copper sulfate, and group third received 20 mg per kg copper sulfate throughout the 16 week. After the 16 week of exposure, we did the behavioral analysis and then finally sacrificed the animal for further biochemical analysis. So here is the data. Animal body weight, you can see here the control animal body weight throughout the 16 week of period, but we found that the copper exposure reduces the body weight in both the group. But however, we have not got any significant result in body weight. Then we measure the copper level. As we export animal to the copper, we found that the copper level was significantly higher in 20 mg per kg group, and the ceruloplasmin, which is considered as a surrogate marker for copper toxicity, it was significantly decreased. So from these copper and ceruloplasmin estimation, we have confirmed that this copper exposure causes the toxicity. Then we evaluated the behavioral parameters. We did locomotor activity and rotor rod test for neurofunctional assessment. We found that the locomotor activity, you can see here this tracking plot showing that the control group shows the maximum activity, means distance travel, but in copper export it was significantly decreased. It can also see here that this distance travel was significantly decreased in 20 mg per kg copper sulfate group. Simultaneously, this rotor rod test, which is used to investigate the motor coordination property of the animal, and we found that the total run time on the rotor rod was significantly decreased in 20 mg per kg copper sulfate group. We also evaluate other neurobehavioral parameters like memory function parameters. So we did NORT test and passive avoidance test, and we found that the memory function was significantly declined in 20 mg per kg copper sulfate group, and other neurobehavioral parameters like elevated plasmase, which is a marker of stress, and immobility time, which is also a marker of anxiety in the animal. So we did four swim test, and we found that the immobility term was higher in the 20 mg per kg copper sulfate group. Then we evaluated the oxidative stress parameter. So here is the question: what is oxidative stress? So oxidative stress is an imbalance between the reactive oxygen species and the antioxidants. So for reactive oxygen species, we measure the nitrite level, A2SDG level, and MDA level. For antioxidant, we measure GSH, SOD, and catalase. So here is the data. You can see here the MDA, nitrite, and 8OHDG level was higher in the copper exposed rat. So we confirmed that our copper exposure increased the reactive oxygen species level. So what is the status of antioxidant enzymes? So we found that the antioxidant enzyme was a decrease in the copper exposed rat, 20 mg per kg copper exposed rat. So from these results, we have confirmed that copper exposure at the higher dose, 20 mg per kg, may lead to the more significant toxicity in contrast to the comparative lower dose. So we have characterized the model, and our next target to see the effect of MiADMSA in this characterized model. So before going to the animal model, we want to confirm whether our drug is binding to the copper or not. So for binding assay, we have performed the fluorescence and NMR spectroscopy. Here is the result. You can see here we took the spectra of alone MiADMSA, then we added the increasing concentration of this copper, and we found that the intensity of the signal was decreased when we increased this copper. So it may be due to the binding of MiADMSA with copper. The similar pattern you can see here after 1 hour incubation and 24-hour incubation. So this indicates that our drug is not showing any metal redistribution. Metal redistribution means when this complex will dissociate, uh, this fluorescence may be hampered. So we found that after 1 hour incubation and 24-hour incubation, the pattern was similar. So it is showing that our drug is not showing any metal redistribution, which is a major issue with the other metal chelator. We further confirmed binding by the proton and carbon NMR, and you can see here we took the spectra of alone MiADMSA and the complex, and you can see here the SH peak here in proton NMR and the CS peak in the carbon NMR. But when we added this copper into this solution, and you can see here these peaks are invisible in the spectra, which is showing that this phenomena might be due to the binding of copper with SH group and CS group. So that is why these peaks are invisible. So here we have confirmed that our drug is binding to the copper. So our next target to see its effect on the cell line. So we took the neuro to a cell line and then exposed the cells with different copper concentration, and we found that at the dose of 50 micromolar dose of copper, it's showing the 50% of cell viability. Then we incubate this cell line with the MiADMSA, and we found the dose dependent recovery in MiADMSA group. When we increased the dose, the cell viability was significantly increased. We also measured this oxidative stress marker in the neuro 2 cell line, and we found that the ROS level was significantly higher, and interestingly our MiADMSA was decreasing the ROS level and the TVOS level. Further, this GSH and catalase level was also significantly ameliorated in the MiADMSA treated rate, which showing that our drug is reducing the oxidative stress level in neuro 2 cell line. So here we have confirmed that our model is characterized and our drug is reducing the copper level from the neuro to a cell line and also the oxidative stress. So our next target to see its effect on the animal model. So we designed this experiment in which we have exposed the animal to the copper for 16 week or 4 month. Then we randomized into different group and exposed with D-penicillamine and MiDMSA. After the 2 week of exposure, we performed the behavioral studies and then sacrificed for animal for further biochemical histological estimation. So as you can see here, we isolate the brain of the rats and found that the copper level was significantly higher in the copper exposed rat, and the MiDMSA and DPA treatment significantly reduced the copper load from the brain, as you can see here. And you can see here that the we found the more significant recoveries as compared to the D-penicillamine in MiDMSA group. Then we evaluated the ACH activity, but we have not found any significant impact on the brain SEH activity and the serum ACH activity. We performed the several neurobehavioral parameters. As you can see here, the copper exposure decreased the movement of the rat, and the MiDMS and DBA treatment recover this locomotor activity. Elevated plasmase performance was significantly decreased. As you can see here, the movement into the open arm was significantly reduced in the copper exposed rat. However, the treatment with MiDMSA and DPA improved this open arm activity, which indicate that the decrease in stress level in copper exposed rat. Then we did the passive avoidance test for memory functions, and we found that this copper exposure caused the significant memory dysfunctions, and treatment with MiDMS and DPA improved the memory functions, but we did not get any significant improvement. Then we evaluated this amyloid beta protein and tau level using the ELISA technique, and we found that the copper exposure significantly increased this amyloid beta and the tau level in the copper exposed rat as compared to the control, and the treatment of MiDMSA and the DPA decreased the A beta level and tau level, which might be due to the decrease in copper level from the brain. As we have seen in the metal estimation results, we found that this MiDMS and DPA decrease the copper level from the brain. Then we also measured the genetic expression of this amyloid beta protein and phosphorylated tau, and we found that our treatment reduced the expression of these proteins. As you can see here, the amyloid beta protein, tau proteins, and the caspase 3 using QRT-PCR method. Then we evaluated the other brain oxidative stress parameters, and we found that you can see here the ROS level and nitrite level, the A2SDG level, and the MD level was higher in the copper exposed rat, and the MiDMSA treatment decreased the oxidative stress marker, which indicate that our drug is reducing the copper load and also decreasing the oxidative stress marker. Simultaneously, the antioxidant enzymes level was also improved in the MiDMSA treatment group, as you can see here. Then we perform the brain histology, and you can see here the normal cellular architecture in the control group and the copper group. As you can see here, more perforated and multinucleated, and this necrotic cells are visible, as you can see here. And this MiDMSA treatment and DPA treatment improved this condition, as you can see here. So our drug is reducing the copper load from the brain. It is also improving the neurofunctions and decreasing the oxidative stress marker, and also the associated protein like A beta and tau protein. So here we can summarize that this MiDMSA prevented from neuronal degeneration, apoptosis, and normalizing the amyloid beta and tau level in the brain. So here we have confirmed that our drug is reducing the copper level from brain. So our next target to see its effect on the this hepatic level. So we designed this experiment similar experiment was designed, and we isolate the liver for further biochemical and histological estimation. So as you can see here, the liver copper level was significantly decreased in the MiADMSA group. So our drug is reducing from copper level from brain as well as from liver, which is a major target in the Wilson disease. These serum parameters, as you can see here, ALT, AST, cholesterol, LDL level was also improved in the MiADMSA treated group as compared to the control. This oxidative stress marker, as you can see here, the marker was significantly higher in the copper exposed rat, and MiADMSA treatment reduced the oxidative stress marker and improved the quality of these antioxidant enzyme. Further, we also measure this hepatic markers. As you can see here, inflammatory markers was also as reactive oxygen species generation causes the inflammatory cascade, initiate the inflammatory cascade. So we also measure these inflammatory markers, and we found that our drug is significantly reducing these markers like IL-1 beta, TNF alpha level, IL-6 level, IL-4 level, and caspase-3 estimation. So our drug is reducing the copper level, also ameliorating the oxidative stress marker, and also these inflammatory markers. The histology study was also performed to evaluate the cellular changes. As you can see here, the normal architecture of this liver cells, and it you can see here the copper exposure causes the degeneration of hepatocytes, and this alignment, sinusoidal alignments, also this portal vein disturbance, central vein disturbance, and the treatment MiADMSA significantly improved the cellular architecture, so which indicate that our drug is improving cellular level as well. The picrocereus red staining was used to evaluate the collagen deposition. As you can see here, there is a minimum collagen deposition, but in copper exposed rate, as you can see here, this red color is indicating the more copper, more collagen deposition, and our treatment MiADMSA significantly ameliorate this collagen deposition, which might be due to the decrease in copper load from the liver, which decrease the oxidative stress marker and also the inflammatory marker, so that it might be due to the reduction of oxidative stress marker and the inflammatory marker. So here I have summarized that copper could be a potential hazardous to the health and may be a risk factor for neurological disorders. Chelation therapy using thiol could be effective in the management of neurological disorders. And last, MiADMSA might be a drug of choice in the management of Wilson disease. However, more detailed studies are required using specific Wilson model to broaden its therapeutic use or for the clinical application. So here I want to acknowledge my supervisor and Department of Pharmaceuticals. And last, I would like to thanks ISF Moga for inviting me at this forum. Thank you, ma'am.
Thank you very much, sir, for your wonderful talk. Now the session is open for discussion. Sir, we have some MPhil students with us. If any of you want to ask anything.
Ma'am, I have a question.
Yes, Simran, you can ask. You can switch on your
मैम
camera.
मेरे को ये पूछ कैमरा
Hello?
भी ऑन करना है?
Yes, you can.
गुड मॉर्निंग सर.
Yes, you
गुड
can.
मॉर्निंग मैम.
Good morning.
सर मेरा क्वेश्चन ये था कि जैसे किसी को विल्सन डिसीज है तो सर्जरी एक ऑप्शन हो सकता है उसको ट्रीट करने के लिए. आप कह रहे थे कि पर्टिकुलर ऑर्गन में इसकी ज्यादा वो पॉइजनिंग होती है, जैसे लीवर आपने बताया, ब्रेन बताया. तो लीवर अगर हम ट्रांसप्लांटेशन करेंगे तो उसकी रिकवरी हो सकती है इस कंडीशन में?
ट्रांसप्लांटेशन इज अ लास्ट ऑप्शन. मतलब जब लीवर डिजनरेट हो जाता है तो ट्रांसप्लांट ही करना पड़ता है विल्सन के केसेस में.
सर उसमें दोबारा डिसीज होने के चांसेस होते हैं कि नहीं होते हैं? लीवर हम ट्रांसप्लांट भी कर दे तो
होने के चांसेस होंगे, क्योंकि ये जेनेटिक डिसऑर्डर है.
तो सर
तो
अगर हमने ट्रांसप्लांट ही कर दिया तो मे बी चांसेस कम भी तो हो सकते हैं.
बट होल लीवर हम ट्रांसप्लांट करें या फिर हम जीन थेरेपी वगैरह के लिए अप्रोच करें तो इट विल बी इफेक्टिव, अदरवाइज तो नहीं होगा.
ठीक. तो सर और इसका मतलब ये कौन सी एज ग्रुप में ज्यादा इफेक्टिव होता है, और मतलब किस एज ग्रुप में ज्यादा इसका प्रेवलेंस है इस डिसीज का?
चाइल्ड्स में ही देखा जाता है सबसे ज्यादा. चाइल्डहुड में ही
ठीक है।
ये प्रोमिनेंट होती है।
पर सर ओके, मतलब ओल्ड एज में इसका कोई कंसेप्ट नहीं है,
ओल्ड
तो
एज
सर
में
फिर
नहीं।
जो भी न्यूरोलॉजिकल डिसऑर्डर्स हैं वो मतलब बच्चों से ही स्टार्ट हो जाता है?
नहीं नहीं, ऐसा नहीं है, मतलब जैसे कि बहुत सारे फैक्टर्स होते हैं, मतलब जैसे कि न्यूरोलॉजिकल डिसऑर्डर्स एक डिफरेंट कंडीशन है। मतलब जैसे कि अल्जाइमर की बात करें हम, तो अल्जाइमर केसेस में क्या होता है कि जो एमिलॉइड बीटा प्रोटीन का एक्सप्रेशन भी हायर होता है, और जब एजिंग होता है तो वहाँ पे क्या होता है कि जो कॉपर ट्रांसपोर्टर होते हैं, कॉपर ट्रांसपोर्टर का एक्सप्रेशन भी हायर हो जाता है, जिसके कारण कि क्या वहाँ पे कॉपर लोड बढ़ जाता है ब्रेन में। सो कॉपर लोड भी बढ़ गया और यहाँ पे आपका अमिलॉइड बीटा प्रोटीन भी ज़्यादा है, तो वो प्रेसिपिटेट होना स्टार्ट कर देता है, तो वो एज कंट्रीब्यूट करता है अल्जाइमर डिसीज में।
ओके तो इसका होम रेमेडीज, अगर हम अपनी बॉडी में कॉपर की एक्यूमुलेशन को कम करना चाहते हो तो कोई ऐसी होम रेमेडीज हो तो?
होम रेमेडीज में एक तो सबसे पहला हम कॉपर रिस्ट्रिक्टेड डाइट यूज कर सकते हैं, मतलब जिनमें मिनिमम कॉपर हो। और सेकंड एक मोरिंगा ओलिफेरा होता है, मतलब सहजन जिसको शायद हिंदी में बोलते हैं, वो हम यूज कर सकते हैं, वो एज अ मेटल किलेटर उसमें प्रॉपर्टी होती है, तो एक वो भी हम यूज कर सकते हैं। और जो जैसे
ओके
एंटीऑक्सीडेंट होते हैं, जैसे कि ग्लूटाथियोन हो गया, उसमें भी थायल ग्रुप होता है, तो थायल ग्रुप भी जो होता है कि कॉपर को क्लेट करता है, आर्सेनिक को भी क्लेट करता है, मतलब जो मेटल को क्लेटिंग प्रॉपर्टी होती है, तो जो एंटीऑक्सीडेंट होते हैं उनसे भी हम ट्रीट कर सकते हैं।
ओके, थैंक यू सर।
Thank you, Simran. Anyone else?
थैंक यू मैम।
Okay, thank you very much, sir, for sharing your valuable research experience with us, especially on the copper toxicity in the Wilson disease and the therapeutic potential of the novel component which you have used in the treatment of neurodegenerative disease. And I hope the knowledge which is shared by you will motivate the student to work more on the metal toxicity in neurodegenerative disorders. So we really appreciate your time and efforts, sir, and thank you very much once again for giving us your valuable time.
थैंक
Thank
यू
you
मैम।
very much.